Welcome to video series on protein metabolism. This video is on metabolism of methionine and cysteine. You will learn in this video. So you will be able to understand for homocystinuria using flowchart, clinical correlation, important MCQ discussion, probable university based examination and memory tricks for quick. Why is methionine and cysteine metabolism important? So you need to understand this from two different perspectives, from the exam point of view and the other one is from patient point of view. From exam point of view, these are the probable questions that can be asked in your internals or in your university examination, and these include enumerate compounds derived from methionine, describe synthesis of cysteine from methionine, explain folate trap, discuss various homocystinuria. And from the patient point of view, there is probability of you encountering congenital homocystinuria, which are actually rare, but you can encounter in pediatric ward. Acquired homocystinuria are more common, and you will encounter these cases in medicine ward. And Garrett's tetrad, again the Garrett's tetrad, cystinuria, cystinosis, all these are rare inborn error of metabolism, which you might encounter during your pediatrics. Now coming to metabolism for methionine, this is usually asked as a long essay. It can be asked as a case based long essay question or a direct long essay question for 10 marks. Now methionine is essential amino acids, so the terminology essential means it has to be consumed in the diet. So the dietary methionine is acted upon by methionine adenosine transferase, where ATP donates the adenosine group to methionine, and ATP gets converted to inorganic pyrophosphate represented as PPI and inorganic phosphate represented as PI, which drives the reaction forward. Methionine gets activated to S-adenosyl methionine. So this is the active form of methionine. In the next step, S-adenosyl methionine gets converted to S-adenosyl homocysteine, and this SAM, S-adenosyl methionine, is abbreviated as SAM, and it is the methyl group donor. It donates the methyl group to any acceptors inside our body, and these acceptors get converted to methylated acceptors. In the next step, adenosyl group is lost from S-adenosyl homocysteine and resulting in conversion of serine and homocysteine. Now this homocysteine can be reconverted back to methionine with the methyl group donated by methyl cobalamin, which gets converted to cobalamin, and the enzyme is methyl transferase. Now this methyl cobalamin actually is the transport form of vitamin B12. It is regenerated back using methyl group donated by tetrahydrofolate, and after donating the methyl group to cobalamin, this methyl tetrahydrofolate get converted to tetrahydrofolate, and this is the active form of folic acid, and the enzyme remains the same. It is also methyl transferase. So in the subsequent step, what happens is homocysteine get converted to cystathionine. Enzyme is cystathionine synthase, and this requires B6 as coenzyme. The active form of B6 is pyridoxal phosphate. So other reaction where PLP acts as coenzyme is the transamination reaction and also glycogen phosphorylase. These are the reaction where PLP acts as coenzyme. Now there is a difference between synthase and synthetase. Synthase are those enzymes that does not require ATP to drive the reaction forward, whereas synthetase requires hydrolysis of ATP to drive the reaction forward. So next, in the next step, cystathionine gets converted to homocysteine by cystathioninase enzyme. Along with homocysteine, cysteine is also formed. This is how cysteine, a non-essential amino acid, is synthesized from methionine. So in the next step, homocysteine undergoes deamination and decarboxylation non-enzymatically to form propionyl CoA. Now this propionyl CoA undergoes carboxylation to form methyl malonyl CoA, which gets converted to succinyl CoA. Now this will enter TCA cycle. Now this succinyl CoA gets converted to succinate, fumarate, malate, oxaloacetate, and this gets converted to pyruvate, and pyruvate by gluconeogenesis gets converted to glucose. This is the reason why methionine is glucogenic. So just to summarize, methionine is a essential amino acid. It is glucogenic. It is involved in synthesis of creatine, which is a non-essential amino acid. And one of the main function of methionine is to participate in transmethylation reaction, where its active form S-adenosyl methionine acts as a methyl group donor. Now sometimes this list all the transmethylation reaction can be asked as a 5-mark question for you. So you need to write all this transmethylation reaction. where guanidinoic acid accepts a methyl group and gets converted to creatinine. So this has been explained in detail in the previous video on glycine metabolism, so you can watch that video. Histidine undergoes methylation to form methyl histidine. Serine undergoes methylation to form choline. So this is how they are involved in synthesis of phospholipids. Phosphatidylserine can get converted to phosphatidylcholine, both of which are phospholipids. They are component of biomembrane. Acetyl serotonin undergoes methylation to form melatonin. Melatonin is responsible for maintaining of our circadian rhythm and biological rhythm, whereas serotonin acts as a neurotransmitter. Ethanolamine undergoes methylation to form choline. Again, phosphatidylcholine is nothing but lecithin, which acts as lung surfactant. So norepinephrine undergoes methylation to from epinephrine. Epinephrine is nothing but adrenaline. It is a hormone synthesized from the adrenal medulla, responsible for fear, flight and fight reaction. Similarly, non-epinephrine can undergo methylation to from metanephrine, which gets excreted in urine. So the other example is carnosine undergoes methylation to form anserine. Both are polyamine, responsible for maintenance of cell growth and differentiation. Apart from this, DNA and tRNA can also undergo methylation. So tRNA, when it undergoes methylation, it facilitates protein biosynthesis. So it is methionine tRNA, which is the first amino acid that is incorporated in during protein synthesis among eukaryotes, and it is formyl methionine tRNA, which is the first amino acid that will be incorporated in protein synthesis in eukaryotes. So this is the most common viva question asked, so you need to remember this. So what happens when the DNA bases are methylated? We know that there are 4 DNA bases, adenine, guanine, cytosine and thymine. When they are methylated, so that particular gene will not be expressed, and this is how gene silencing occurs inside the body. Now coming to disorders associated with methionine metabolism. So these are known as homocystinurias. Now this homocystinurias, what are the causes for this homocystinurias? Now just to briefly trace the pathway. So methionine gets converted to SAM, and then this gets converted to estradiol homocysteine, gets converted to homocysteine, and this is acted upon by cystathionine beta synthase enzyme, which forms cystathionine. Now when this enzyme is deficient, it results in classical homocystinurias. Apart from that, acquired homocystinurias occur due to deficiency of B12, folic acid and B6. Now as you all know, B12 is required for conversion of homocysteine back to methionine, and methylcobalamin is generated back with the methyl group donated by tetrahydrofolic acid. So when folic acid is deficient and when B12 is deficient, there occurs accumulation of homocysteine, and this gets excreted in urine as homocystinuria. And PLP acts as a coenzyme for cysteine synthase. So when this enzyme is deficient, again there occurs accumulation of homocysteine and it can result in homocystinuria. What are the characteristic features of this homocystinuria? Now this accumulated homocysteine can interfere with collagen cross-linking, and as you all know, collagen is a ground substance and is one of the important component of extracellular matrix. So when this involves the eyes, especially where the lens is located, this can displace the lens and result in lens dislocation. And when this collagen cross-linking interferes with the bones and joints, the gait of the child can be altered and resulting in Charlie Chaplin gait. Now, so the second harmful effect of accumulation of homocysteine is it activates factor 12, that is Higman's factor. This stimulates platelet aggregation, thrombus formation, and this thrombus can be formed in medium-sized vessel and large-sized vessels of heart, resulting in atherosclerosis and MI, and in cerebral circulation. If it is affected, it can manifest as stroke. The other deadly effect is homocysteine causes oxidation of LDL by forming a free radical called homocysteine thiolactone, and this oxidized LDL is taken up by macrophages, get converted to foam cells, and foam cells predispose to formation of atherosclerosis, and which later can manifest as myocardial infarction. Now, how to diagnose this condition? There is increased homocysteine in blood, increased homocysteine in urine, and there is one simple screening test to identify homocystinuria, that is cyanide nitroprusside test. So urine sample is collected and sodium nitroprusside, when it is added, it forms a magenta red colored complex. So normal reference interval for homocysteine is 5 to 15 micromoles per liter. When there is 5 millimole per liter increase in homocysteine, this is equal to 20 milligram per deciliter increase in cholesterol. Okay. So now just a quick summary of homocystinuria. Two types, classical or congenital due to deficiency of cysteine beta synthase enzyme, acquired due to deficiency of folic acid B12 and pyridoxal. Characteristic feature, thrombosis due to activation of factor 12, atherosclerosis due to oxidation of LDL, and because of interference with collagen cross-linking, there occurs Charlie-Chaplin gait, lens dislocation and long extremities. It can be identified as a screening test, that is sodium nitroprusside test or the cyanide nitroprusside test, where urine changes its color to magenta red colored. Now one more concept that is associated with the methionine metabolism is folate trap. Now what is this folate trap? For the conversion of homocysteine back to methionine, methyl group is needed, and this is donated by methyl cobalamin, which becomes cobalamin, and methyl cobalamin is regenerated back with the methyl group being donated by tetrahydrofolic acid. So the active form of tetrahydrofolic acid is generated. So when there occurs deficiency of B12, so what happens is the active form of folic acid is not generated. So this is folate trap. Now why we should worry about this trap? So deficiency of folic acid results in megaloblastic anemia because folic acid is responsible for RNA maturation. DNA synthesis is inhibited, but protein synthesis continues and the RBC become megaloblast. And as you all know, DNA is required for the differentiation of neural cells. There occurs neural tube defects in folic acid deficiency. And as a person is having B12 deficiency, there occurs accumulation of homocysteine, which predisposes to thrombus formation, platelet aggregation, and causes oxidation of LDL and predisposes the individual to atherosclerosis. Next, coming to metabolism of cysteine. Cysteine is a non-essential amino acid which is synthesized from methionine. It is glucogenic because cysteine undergoes transformation to form beta-mercaptopyruvate, which gets converted to pyruvate, and pyruvate by gluconeogenesis gets converted to glucose. Coming to functions of cysteine, cysteine forms disulfide bridges to make the protein active. Cysteine forms taurine. How this is the mechanism by which cysteine forms taurine. Cysteine undergoes oxidation to form cystic acid. This undergoes decarboxylation to form taurine, and taurine is used for conjugation of bile acids. Now these bile acids are derived from cholesterol. Cholesterol gets converted to cholestyne, diol and triol, which forms folic acid and quinodeoxyfolic acid. These are conjugated with tyrine to form bile salts. And these bile salts are used for absorption, digestion, absorption of fat soluble vitamins and lipids. So glycine is also another conjugating agent. Check out the video on glycine metabolism for formation of bile salts and bile acids. Then cysteine also forms glutathione. Now this glutathione is a tripeptide made up of glycine, glutamate and cysteine. Glutathione function itself can be asked as a 5-mark question in your examination. Coming to functions of glutathione. Glutathione is involved in conversion of methemoglobin to hemoglobin. Methemoglobin is a state where iron is present in oxidized form, that is ferric form. Ferric form is reduced to ferrous form with the help of glutathione reductase enzyme, so that hemoglobin gets its oxygen carrying capacity back. The next important function, it is involved in transportation of neutral amino acids through Meester cycle. So you can check out this video, the first video on digestion absorption of proteins, where the Meester cycle is explained. Now just to give you the bird's eye view of Meester cycle, neutral amino acid combined with glutathione to form glutamyl amino acids. Amino acids are released into general circulation. Glutamate gets converted to oxoproline. This is acted upon by oxoprolinase enzyme to form glutamate. Glutamate combines with cysteine to form gamma glutamate cysteine. Later on, it combines with glycine to get to regenerate back the reduced glutathione. This is how glutathione is involved in transportation of neutral amino acids like phenylalanine, tyrosine, tryptophan, valine, leucine, isoleucine from small intestine into the blood circulation. Apart from that, glutathione is involved in conjugating heavy metals, therapeutic drugs and organophosphorus compounds. So it acts as a conjugating agent. So the last important function of glutathione is it is involved in maintenance of RBC membrane integrity. So many free radicals are generated inside the RBC. So one such powerful free radical is superoxide, which is chelated by superoxide, dismutase to form hydrogen peroxide. Now hydrogen peroxide itself is a powerful and harmful free radical. So this is safely reduced to water by glutathione peroxidase, during which time the reduced form of glutathione GSH gets converted to oxidized form represented as GSSG. Anything in oxidized form is always dangerous to the body, so this has to be reduced back with the help of NADPH, who donates the hydrogen atom to oxidized to glutathione to gets converted to reduced glutathione, and NADPH gets oxidized to NADP. NADPH comes from HMP pathway. So just to summarize cysteine metabolism, cysteine is a non-essential amino acid. It's glucogenic because it forms pyruvate, forms disulfide bridges in proteins, forms taurine, which is used as a conjugating agent for bile acids, form glutathione. Glutathione itself can be asked as a 5-marker question. So this is a quick summary of glutathione. It's a tripeptide made up of glycine, glutamate, cysteine. Functions include conversion of methemoglobin to hemoglobin, acts as a conjugating agent, maintain RBC membrane integrity, and transports nucleic acids. Coming to other clinical significance, cystinuria and Garrot's tetrad. Now, what's the biochemical defect here? There is mutation in the amino acid transport protein involving the proximal convoluted tubules of the kidney. So this affects the transportation of dibasic amino acids, cysteine, ornithine, arginine, chol, arginine, lysine, and the mnemonic to remember these four is chol. C stands for cysteine, O stands for ornithine, A stands for arginine, and L stands for lysine. All these will be excreted in urine, and these are the components of Garrett's tetrad. Garrett was the scientist who identified the disorder where there is excretion of cysteine, ornithine, arginine, and lysine. Now, cysteine is poorly soluble in urine. It gets precipitate and manifests as kidney stones. Only cysteine precipitates and not the other dibasic amino acid. This is because ornithine, lysine, arginine are soluble in acidic urine, whereas cysteine is poorly soluble and precipitates as stones in the form of hexagonal crystals. What are the clinical features? So child or the adult usually presents with the formation of recurrent kidney stones, flank pain. This is a flank is the area where anatomically kidney is located. Hematuria, because blood is excreted in urine, and this kidney stones they interrupt with the blood supply of the kidney, and they damage the blood vessels of the kidney. Rather than interrupting, they damage the blood vessels, and urinary tract obstruction because the crystals can obstruct the flow of the urine. How to diagnose this condition? So when urine is observed under microscopy, hexagonal crystals are seen. Cyanide nitroprusside test is positive with the development of magenta red colored urine. Then urine cysteine levels are. So the other clinical significance or the correlation condition is cystinosis. It's an autosomal recessive disorder where the crystals are accumulated in lysosome. It's a lysosomal storage disorder. Just to recall the function of lysosome, protein degradation takes place in lysosome where the protein is broken down to individual amino acids, and these amino acids are released into general circulation so that they can participate in translation process where protein biosynthesis can take place. Here in cystinosis problem is cysteine does not come out of lysosome, stays back inside the lysosome in multiple organs, manifesting as cystinosis. There are 3 important types of cystinosis. These include the infantile or the nephropathic cystinosis, most common and severe. This is closely associated Fanconi syndrome. Now, Fanconi syndrome is a condition where mainly affects the kidneys. The proximal convoluted tubule is affected. Normal function of kidneys, the whatever the glomerular filtrate appears in the proximal convoluted tubule, 80% of it gets reabsorbed back into the circulation, or this becomes defective. So glucose, amino acids, phosphate, and bicarbonate, they are not reabsorbed back at the level of proximal convoluted tubule, and they are excreted in the form of glycosuria, amino aciduria. Here cysteine is also excreted in the form of cysteinuria, phosphaturia, bicarbonate loss, manifesting as metabolic acidosis. Because all these are excreted and there is deficient energy because glucose is excreted, there is failure to thrive, no amino acids, no building block, phosphate is excreted, child will have rickets, and this will pose a very huge load on the kidney, so kidney starts failing. The other form is juvenile form. This is milder form and compared to the infantile form, and there is mild renal impairment. The third form is the adult form. Kidneys are spared, on the eyes are involved, where the cysteine is crystals are accumulating in the cornea. So coming to the lab diagnosis, so there will be increased cysteine in the leukocytes, and the adult form, when the eyes are examined using slit lamp examination, the crystals are seen in the Yeah, so to sum up, this was all about the metabolism of methionine and cysteine. To sum up, so these are only the key features, and you can use these tables for your quick recall purpose. Methionine, essential amino acid, it is glucogenic because it forms a propyl CoA which gets converted to succinyl CoA, and this enters TCA cycle, forms oxaloacetate pyruvate, and pyruvate gets converted to glucose. Synthesis of cysteine is done by methionine, and one of the important function of methionine is participation in transmethylation reaction, where it acts as methyl donor through SAM, S-adenosyl methionine. Coming to the clinical significance part, homocystinuria, two types, congenital or the classical due to deficiency of cysteine synthase enzyme, acquired due to deficiency of folic acid, B12 and pyridoxine. Acquired is more common when compared to the classical form. Characteristic feature of homocystinuria is homocysteine, accumulated homocysteine activates thrombosis by stimulating factor 12, predispose atherosclerosis by oxidizing LDL, and by interfering with collagen cross-linking, alters the gait of the child in the form of Charlie Chaplin's gait, lens dislocation and long extremities. So there is a screening test to identify homocystinuria, that is cyanide nitroprusside test, wherein the urine forms magenta red color. Coming to metabolism of cysteine, it's a non-essential amino acid synthesized from methionine. It is glucogenic because it forms pyruvate, and pyruvate can get converted to glucose by gluconeogenesis. Forms disulfide bridges in proteins, forms taurine, which is used as a conjugating agents for bile acids, cholic acid and chenodeoxycholic acids, forms glutathione. Glutathione itself can be asked as a 5-marker question. It's a tripeptide made up of glycine, glutamate, cysteine. Functions of glutathione includes converts methemoglobin to hemoglobin by reducing ferric form of iron to ferrous. Conjugating agents for heavy metals, drugs and organophosphorus compounds, maintains RBC membrane integrity and transports neutral amino acids using cycle. So this was all about metabolism of methionine and cysteine. Coming to self-assessment, this will help you to identify your areas of weakness and your areas of strength. So the immediate activated form of methionine used in methylation reaction is 3,2,1, and the answer is S-adenosyl methionine. The enzyme deficient in classical homocystinuria, 3,2,1. Answer is cysteine beta synthase involved in conversion of homocysteine to cysteine. Conversion of homocysteine to methionine requires 3,2,1. Answer is universal methyl donor in the body is 3,2,1, and the answer is S-adenosyl. Elevated homocysteine levels are associated with 3,2,1, and the answer is thrombosis because homocysteine activates factor. Which vitamin is required for the conversion of homocysteine to cystathionine? 3,2,1, and the answer is B6. PLP acts as a coenzyme for cystathionine beta synthase. In vitamin B12 deficiency, which metabolite increases? 3,2,1. Answer is homocysteine, because for conversion of homocysteine back to methionine, B12 is S-adenosyl methionine is formed by addition of 3,2,1. Answer is adenosyl group donated by P. Which pathway links methionine metabolism with folate metabolism? 3,2,1, and the answer is 1-carbon metabolism, which is involved in carrying the methyl group from tetrahydrofolate. A patient with marfanoid habitus, lens dislocation downward and thromboembolism likely has a defect in, answer is cystathionine beta synthase. So this was all about the self-assessment part. Score yourself. If the score is more than 50%, you can repeat watching the video and redo all the MCQs after 24 to 48 hours. If your score is less than 50%, so it is better you redo the entire thing immediately. So thank you so much. Watch the playlist on protein metabolism for complete clarity. I'll be uploading one video after the other in this playlist. So the next video will be on phenylalanine and tyrosine metabolism. Thank you all. Happy learning. Please do like, share and subscribe.